I don't know why I'm here, but maybe Aldo and Stephen do. I mean, I'm not even really an economist. I just play one on TV. But I'm definitely not a geneticist. But what Aldo suggested might be useful would be to talk about an issue that's arisen in the last decade and a half at the level of cellular neuroscience. And that is that cellular neuroscientists are less and less convinced that the gene by environment distinction, or genes and environment distinction, is going to be useful to biologists. And so what I was going to do is just tell you the story that led to that worldview that's real common amongst systems cellular level neurobiologists. And so this is just a story. I could tell you about 10 stories. They all come out the same way. But this is kind of the most beautiful one. OK, so to tell this story, I'm going to tell you about the apparently genetically determined structure of the human visual system. And so you're looking at a cross-section through a human brain. And for the moment, I just want you to admire this architectural beauty of this object that I'm going to tell you a story about. So these are your two retinas, your two eyes. And they project back through a series of relays to the back of your head to the visual cortex, which is where the first stage of the brain's representation of the visual world occurs. And these pathways are always the same in every one, and they're incredibly stereotyped in their appearance. It actually turns out that the details of these pathways are incredibly precisely stereotyped. So here we're kind of looking at the 4 different quadrants of a retina, and I want you to get the idea that the yellow part of the retina is projecting here. The blue part of the retina is projecting to here. What it actually is there's a topographic mapping rule. So every point on the retina projects to a matched point in the visual cortex. And you can think of the visual cortex as forming, here you can see this a little better, a map of the outside world. So what you want to think of is that you take the retina, and it's actually an affine transformed projection. It's kind of log expanded so that the center of vision consumes more representational space than the periphery. But in any case, there's this really precise point-to-point topographic map of the retina, and hence the outside world, lying in the visual cortex. And this has been known for a long time. And certainly since the 70s, we've known that if we zoom in on a little point in that map, so you can think of this map kind of as representing loosely a pixel in the world. If we zoom in on the chunk of brain that represents a pixel or a few pixels in the world, we see lots more structure, and it looks genetically determined because we all have exactly the same structure. And here what I'm showing you is a famous experiment by Hubel and Wiesel. And what these guys did was inject a radioactive compound into the eye of, unfortunately, a baby cat. And that compound propagates backwards to this part of the brain. And in blue, you can see the things that were connected to the injected eye. And in beige and tissue color, the things that are connected to the other eye. The thing I want to get across is that each pixel actually in the brain, this is the whole visual cortex laid out flat, alternates its representation of the two eyes. So here's the right eye looking out at space. Here's the left eye looking out at space. And so this little patch right here has a right eye and a left eye looking out at space. Everyone good with this idea? In fact, it's a nice smooth function. This makes it look like a discrete function because it's been thresholded so that cells at the border are kind of looking out of both eyes. OK, so that's cool. These guys took this the next step, and they discovered that there were other classes of information, representation laid out in these pixels. So now we're looking at a bunch of pixels. One pixel might be about that big. And in color, what I'm telling you is what the cells at this location are most sensitive to with regard to an oriented bar in the world. So cells at this location are interested in bars oriented this way. Cells at the red location are interested in bars oriented this way. Everyone get this idea? So there's this smooth, progressive representation of the orientation that a stimulus is at. Now, I'm skipping. This is kind of the way I would teach it to undergraduates. I apologize. The math is so beautiful, but it would take a while. You can actually prove that these things aren't looking for bars. What they're actually doing is spatially restricted Fourier decompositions. And you're getting the main axis of a two-dimensional Fourier decomposition here. And I'm just telling you that as a bar. But the math is really beautiful, but not the point here. OK, so if I go to any one of your brains, and I zoom in, well, if I zoom out, I'll notice a perfect topographic map of the outside world, where the point-to-point topography, less this affine transform, is completely robust. I can predict where in Dalton's brain I'll find me, as long as I'm standing here and Dalton's standing there and looking at me. And at each point in that map, this is about a 2.5 millimeter by 1.25 millimeter slug of Dalton's brain. This left-hand side has representing one eye. The right-hand side is representing the other. As we move across, the likelihood that we'll be drawing information from the eyes varies as a smooth function. And right here, all the cells are binocular. As we move in a spiral around, indicated by color, the main axis of the Fourier decomposition is rotating. And it actually turns out that the phase angle of the Fourier decomposition is also stepping in a lawful manner. And these blocks in the middle are actually lawfully representing color information. So outside of those blocks, the cells are only interested in black and white contrast. And inside of those blocks, they represent color also in a very precise mosaic form. And when this stuff was first discovered, I think there was a lot of head scratching about this, because this is an awful lot of structure, and there aren't an awful lot of genes. That wasn't clear at the time. When this was first discovered, we thought there were going to be 300,000 genes in the human genome, and people were kind of open to the idea that you probably used a lot of genes to do this patterning, that those genes would be specific to patterning this object. But as it became clear that the number of genes sitting around to be used was really, really small, the question of how this patterning could work became more and more problematic for neurobiologists. Okay, so that's the story first. And it's really cool, and everybody grew up on it who's a neurobiologist. Now I'm going to take you to a simpler system. It's the exact same story, but we're going to switch to the frog, because this is where the genetic problem was figured out. So now you're looking at the same story. This is a frog's brain. This is a frog's retina. And I've just redrawn the arrow projected onto the frog's retina, onto the frog's visual system in its brain. So you can see there's still a point-to-point affine transform topography. It's in a slightly different place. Obviously, he's a frog. And it represents both axes. Everybody's good with this, right? Okay, so our first question is going to be, how do we think about the building of this topographic map at a genetic level? And Roger Sperry won the Nobel Prize for providing what was originally thought to be the right answer to that question. And what Sperry said was, well, let's think about this. Let's imagine we had some protein which is a marker for the lower half of the retina. Now, what he actually suggested were gradients. So for you, let me say it in a more sophisticated way. Let's imagine there's a gradient in the y direction of some arbitrary compound x. And this compound has a highest concentration here and a lowest concentration at the top. Now, let's imagine that there's a matched compound that goes the other way. So you have two gradients that cross. So you specify a unique location, north to south, by specifying the ratio of these two compounds. So the idea was that when you get back into the brain, so here's the two axes, there'd be a matched set of compounds. So they would also lay a tile of increasing and decreasing concentration. And basically the job of the neuron would be to hunt back to this part of the brain and then hunt around until it found the ratio that it was looking for. And so the way this was imagined to be done was that this would be in the cells coming from the retina, they would express a receptor ratio. The cells here would be expressing on their cell surfaces the ligand or drug or compound that binds to that receptor in a ratio. And so the little neuron would hunt to that location and park. And so the idea was that there would be some set of these compounds, and this would at least solve the topographic map problem, and you'd basically be doing some kind of slightly redundant XY decomposition using known biochemical mechanisms, so it all would make sense. And it doesn't call, if you think about it, for that many genes, both because you don't need that many patterning genes, and because you could use these in other brain systems or other body systems for similar functions. So that's great. So everybody feels happy about that. And let's see, sorry, I have an animation that I didn't use. And to support that, this is one of the most famous nature-nurture experiments in neuroscience. Sperry did this really cool, famous, and weird experiment. Okay, so if a fly is detected by the little, obviously you know this, frogs eat flies. Fly goes by, the frog goes. So here's a fly, and its image is being cast at this location on the retina. Now, if it's being cast on the ventral retina, that means the fly is up high. And of course, where it is tells the froggy where to strike. Think about the frog's eyes as not moving, which is to a first approximation true. So you can actually think of this map in the brain as, amongst other things, providing a strike map. It tells you what direction to throw your tongue to hit the fly. And the neurobiology of that system had been pretty well worked out at this point. But so here's this crazy idea that Sperry has. He says, OK, if this is all hardwired, and they were using the word genetics at the time, but if it's hardwired, then you could do this crazy thing. You'd go to the frog, you'd anesthetize him, you'd cut the eyeball loose while leaving the optic nerve in place, you'd rotate the eyeball 180 degrees and glue it back in, well, or sew it back in. And now the ventral retina has been rotated to the dorsal surface. So if a fly's up here, the frog should strike down there. And fantastically enough, this works. The frogs do it. It's really cool. Because the frog's eyes, this is important, are non-binocular, that one looks to the right, one looks to the left, you can actually get him able to strike on one side, strike correctly on one side, and strike upside down on the other. And Sperry, following in the line of his mentor, who was a very important neurobiologist by the name of Paul Weiss at Rockefeller, Sperry spent a lot of time trying to train these frogs to sort of correct this problem and showed that the frogs were completely aplastic. The frogs will starve to death long before they'll figure out that they're striking in the wrong direction. So this was taken as evidence that there were no environmental interactions in this story and that this was a genetic story. And really, it's a story about no plasticity, but in 1970, this was seen as no plasticity implying genetics. OK, so now what I want to do is now take apart the biochemistry of how this thing gets built. And it's going to turn out to be very surprising, I think. Before I do that, I'm going to show you a couple of movies, because Aldo asked me to. And so the first thing we're going to do is I'm going to show you a movie of what happens as a cell grows into the optic tectum, the visual system of the frog. And you're just going to see a time lapse of a half day of 2 neurons growing out of the retina. I don't even know why I'm showing this, but Aldo thought it was super cool. Is it starting? Yeah. So here, that's a slice. This is a frog's cortex, a frog's brain. And you can actually see these 2 neurons chasing down towards where they're going to go live. I want you to notice their tips, which are puffballs. And you notice that their puffballs get a little bigger as they get to their target. And the idea is that they're biochemically sniffing for their target chemicals to park them in the right place. OK. So now you can see what actually happens when they find the right place. This is from a different system, but this is what it looks like when the puffballs find the right place. So this cell, you can probably tell, was hunting for this cell. This is laid out in a dish, so it's a really toy problem. And you can see that, I'm going to show it again in a moment. This guy has a big puffball at the end. And once he makes a stabilizing connection with this second cell, he collapses, and they become just annealed to each other. OK, that's cool. Even I think, Aldo, that's super cool. OK, so here was the story that began to emerge. When you look at that, what I just showed you, the cells seem to hunt out to their target location in the affine transform topographic map. Oh, that's terrible. And then they collapse. And a bunch of things immediately began to emerge that were weird. One was, if you raise the frogs in total darkness, you get to this stage, but you never get to this stage. So in the absence of visual input, the thing doesn't seem to work. And it's not just biochemistry or something like that, because if you block electrical activity in these cells, it also doesn't collapse. Frank Schmidt did this very famous experiment where then he raised the frogs in stroboscopic environments. So they could still see, there was still input, but it would only be like. When that happens, all the cells discharge synchronously. So you're changing the correlation structure of the firing rates. And again, you get this behavior. In fact, none of these maps form in any animal under any of these conditions. Now, what emerged was that what was driving the collapse of the map was a process that was basically a mathematical coincidence detection. And I'm not going to tell you the biophysics of how it works, but the critical idea was that there's an ion channel that looks to see whether this synapse that it lives in is busy at a time when its nearest neighbors are busy. And if its nearest neighbors are busy at the same time it's busy, it gets stabilized. And if it's busy at a time when its nearest neighbors are not busy, it dies off. Now, if you think this through, this makes really great sense. Because it means that if you started with a very coarsely tuned map, cells go into into their target and they blow out these big puffballs. And then they make a million connections with all their nearest neighbors. But synapses only get stabilized amongst groups of connections that are highly correlated in their firing rates. Okay, so what are you exploiting here? Why would this work? This would work because two places on the retina that are right next to each other are highly correlated in what they see. And as two places on the retina drift apart, they decorrelate. See, the covariance matrix for the world, for the visual world, is this smooth peaked function. And you can prove that by checking in the world. And so what this mechanism does is it builds the map simply by obeying the rule, I'm looking to minimize the covariance error. I'm looking to maximize covariance between adjacent elements of the topography. That's the only rule. Now, if you think about it, that rule is sufficient to give you everything I told you about. It would, for example, cause patches to anneal that represented the two different eyes because the covariance structure in the two different eyes is a little different. It would cause these kinds of structures, these kinds of structures to arise because all these cells are looking at the same kind of stuff, oriented bars of a certain direction, or the same Fourier component. And so the idea is that what's actually emerging here is the minimum discontinuity map. You're trying to map the outside world, and all you do is you take the covariance structure of the outside world, and you say, oh, I'm going to make the minimum step in variance, and anneal a system that does that, and you get all this structure for free. So now here's a really weird question. I mean, is this genetics? It's an incredibly simple genetic rule that requires just a few genetic components. But it gives a tremendous amount of structure, but all the structure comes from the covariance structure of the real world. If, for example, we were to raise a kitten in a world where there was only television snow, right? So all the pixels are independent. They're all IID. Then you wouldn't get this structure. You'd get a snow cortex. And I mean, this was a big deal. I'll just give one last story because this is so cool. So the person who really nailed this was a Princeton neurobiologist by the name of Martha Constantine Patton, and she said, if this is all true, then I ought to be able to produce kind of any visual system I want surgically. And so what she did is she took a frog, she gave him a third eye. She referred to the frog who gave this up as the donor, which I always thought was cute. And so the donor eye gets plugged in next to the normal eye. And the prediction is, if you understand the variance structure, because the eyes are pointed in different directions, you should get very sharply defined eye dominance representations in the frog, a feature which was believed to have arisen in evolution with the onset of binocularity much later in evolutionary history. And sure enough, I'm not showing her cartoon, you get exactly the same structure you saw in the primate brain, suggesting that that didn't evolve by evolution and evolution of a new mechanism. It just involved piping the input from the two eyes to the same location. And the extant system then, because it's solving this covariance problem, produces this mapping.
Wait a minute, she got exactly the same structure.
Well, so I'm showing you the monkey one because I couldn't find hers last night when you told me to do this.
The same width of the...
No, so that's a good question, is how the width works. And that has to do, if you think about it, that's a resource limitation problem. It has to do with how many neurons you have, right? So you would like to tune the covariance mechanism to respect your total capacity. Think of this as rational inattention. It's like rational inattention.
Who is
So the width.
who is directing this now? There is no.
No, so this is starting. Okay, so the biochemistry for this would be to control the mean and MDA receptor density, and it would be different in different species. Or it could be the receptor affinity, or it could be their the picosiemen conductance of their particular NMDA channel for calcium atoms. I mean, I think there's lots of good mechanisms for understanding how that would work globally. The answer is that the spacing is even different in monkeys and humans. In humans, the spacing is closer to 1.25 millimeters. In monkeys, it's just under 1 millimeter. Okay, but so this is why people got frustrated with the gene environment thing. So is this genes or is this environment? And so a lot of neurobiologists started saying it's an ill-posed question at this level of analysis. You can't disentangle the two. And because the statistics of the visual world are on Earth essentially fixed, people have spent a huge amount of time to demonstrate this. You always get this particular representation out for this particular pair of eyeballs and architecture. Of course, in a world where that wasn't true, you'd get a very, very different system. But so this is why, Jim, before I was talking about this issue of how much variance do you have in environment. This thing's exploiting something for which there is no variance in environment. And so that's why people begin to say, oh, well, so what isn't? This would always show up as genetic. Conditional upon the environment, you don't know it, but the thing that the environment is providing to the system is fixed. Sorry, I took a little too long there.
So why isn't the criterion of choice not itself genetically determined? I mean, I'm not quite, so I understand what you're saying. It's very interesting.
That's fine.
You're okay, fine. You're not, so I understand. You're criticizing this kind of use of standard statistical heritability calculations and so forth. That's what you're looking at.
Yeah. And I think the right way to think about it, obviously, is to think about the statistical structure of the environment and the statistical structure of the genetic constraint and how they interact. And what most neurobiologists working at this level think is, well, that in many cases that's hopeless because the statistical structure of the environment is hidden. And you can surprisingly find yourself in places where the statistical structure of the environment is perfectly constrained with regard to these properties. And you didn't know it for 50 years. And so here you were saying, oh, that's genetic. But I mean, at a mechanistic level, I'm not saying this at a psychological level, but at a mechanistic level, there was nothing really that was coming out of the genetics but the basic topographic map. And not at high resolution.
But that has great survival value, presumably, that you can adapt this way, right?
Well, so you can actually show
over long epics where you have a lot of variance in different situations. It looks like, I don't know what your position is here on that.
No, so there's a normative theory about this, and the normative theory is that essentially what you're doing here is providing the most information you can about what deviates from your priors, right? Because your priors are the map.
Right.
The map is just building your priors into the space. And so you can make the argument that this maximizes information in a finite capacity structure. So the prediction is that if you could, the fundamental visual frequency falls off as 1/f in the real world. Nobody really understands that. And you could imagine a world where that wasn't true, where the distribution of visual frequencies was flat or some other shape. And that animal should have a different visual cortex, right? And we imagine it would. So this is actually an efficient solution. It's a much more efficient solution.
Actually, there was an interesting article in I think it's the International Journal of Epidemiology, that the important point you're making was something that Hogben and R.A. Fisher debated a long time ago as well. That Hogben talked about gene-environment interaction in the way you're talking about it, and Fisher in more an analysis of variance. To me, but maybe not to you, so I'd be interested in your thoughts on it. It seems to me different to talk about the development of the visual system, how our visual systems work, what is essentially common about that, versus, let's take a specific example, why somebody develops schizophrenia and another person does not. nobody's going to say that schizophrenia is genetically determined. It's heritable. And knowing that schizophrenia, I would argue, knowing that it was heritable was a major breakthrough in terms of how to approach trying to understand its neurobiology. Am I wrong? Would you say I'm wrong on that?
No, I mean,
I think it's different to talk about individual differences versus common mechanisms. But
so I mean, I'm pretty schizophrenic on this myself. So if I'm a neurobiologist and a strict structuralist, everything propagates up, then I have one answer, which is kind of this one, which I said more to be difficult and because Aldo enjoyed us talking about it. If I'm being an economist, what I think I do is I say, well, variation in the external world is, the variation I'm aware of in the external world is limited, but I'm going to treat the things that are sensitive to that as environmental variables. So this is kind of a response to Stephen's question. I think about the twin studies as kind of bounding, and I don't really know what they tell me about environment, but they place some sort of soft restrictions on a maximum. And I just live with that number, even though I kind of acknowledge that it's not either structurally or hard and mathematically provably true.
But it seems to me that one thing that a lot of people think about my field, which is behavioral genetics, is that somehow what we're interested in is saying that something is heritable, which is a trivial observation, I would say now, is that it at best tells you something about where you would go next, rather than being an explanation of the behavior. And maybe it's no doubt our fault because we haven't communicated that well. And I think you see that playing out throughout psychiatric genetics, certainly now, is that nobody's interested in the heritability of schizophrenia anymore, even though that was an important observation to get us where we're at today. Whether or not the genetic strategy works in schizophrenia remains to be determined, but it certainly has created that approach, or at least just.
So let me be a tiny bit more aggressive and put it the way I think Cesarini or Dan would.
Mm-hmm.
Cohort effects. Let's say it could only study people who live in Lithuania, and you would come to one set of conclusions about what the distribution of gene-environment interaction was for some disease. And then, because that's just exactly this case, and then I land you on Mars, or worse, in New York, and now you say, oh, conditional on you having just revealed the existence of this new environment to me, I now can pick out more stuff. And of course, you can perform this game indefinitely. Now, these guys point out that you can remove that. I mean, I'm not even sure I'm struggling with is that good? Remove that by doing a principal components decomposition, but then they point out you lose stuff when you do that, of course. And what does that even mean? Like, are you revealing new environmental interactions, or are you controlling for a failure to see the environment as broad enough? This is kind of, I think, in the domain of Stephen's fetching, but I mean, I share it.
Yeah, several years ago, together with the Amsterdam Brain Institute, we did simulations of growing neural networks. And instead of having one, we made MZ and DZ twins. Right. And they started up with a heritability almost 1. And then they perform, and what's essential here is the growth mechanism has a heritability of 1, and the starting factor has a heritability of 1. And then that is put in a simulated environment. And then during the learning, you see that the manifest heritability goes down. So if you look at the output of these neural networks, and you do standard behavior genetical analysis, you see that the heritability goes to 0. But if you estimate, and suppose this was implemented, but if you would estimate the parameters in the growth mechanism, has a heritability of 1 and keeps a heritability of 1. So that shows you that it is extremely important how you define your phenotype here. And I would say here too, this is a kind of correlation mechanism which acts in real time. That correlation mechanism may have a very high heritability, but the output you see here may not. So.
Well, I mean, empirically, for this particular system,
Yeah.
you can't tell people's brains apart from this. I mean, it's true that there's stochasticity in this representation,
Yeah.
but less the fact that there's a little stochasticity in the fine detailed structure. You can, for this particular problem, you can raise a cat in Brussels or in Amsterdam or in New York, and the cat looks the same.
But my only point is that if you sometimes, so in the case of this simulated network behavior, if you look at the behavior and it has been going on for a while, there is hardly any heritability observable. Whereas if you look a level higher at the learning parameters, it is highly heritable.
So, I mean, I think I basically agree with that. I mean, this is a mechanism, I mean, we can all agree that this mechanism that produces these systems, we actually know, where the genes are and their particular receptors. We know they vary. We know about their biophysics. We even know about their thermodynamic properties now. So that mechanism is genetically encoded. It's heritable. We even know its history, recent evolutionary history. And it produces something which looks the same in everyone by exploiting structure in the environment. Now, I mean, that makes really good sense. If you only have 30,000 genes, you have no alternative technique as an engineer but to exploit regularities in the external environment to produce lawful structure. It just has to work that way. But then that tangles with this issue a little bit of what constitutes environment. And I think that we're kind of interested in, I think this is what you're saying, things in the environment that vary, and not things in the environment that are fixed.
Similarly for the genes.
Right, and so we define environment, when we say environment, probably what we mean is things in the environment that vary in a useful way. And that gets confusing when we start talking about cohort effects. Because when we look across cohorts, we have to relax that definition or tighten it, depending on which way you want to think about it, right?
So, let me talk about an issue that might be interesting for economists. Like, suppose I want to see how the way in which I trade off present for future payments is. So I'm now, rather than looking at V1, I'm looking at orbital front.
Mm-hmm.
And now I can think of an experiment which I change the stochastic nature of the reward processes in the world by changing it, making it more frequent instead. Okay. And that presumably then would change the way in which I code present versus future rewards, independently of the individual and his genetic endowment. Is that what you're saying? But do we have any estimates from this study in the visual cortex of how big the change in the environment you have to make so that you can change the structure of the organization?
Okay, so I'm going to unpack that because this is like a conversation Aldo and I have been having for 2 years now. The parts of the brain where you represent, okay, so be economists for a second, and let's all agree that goods have attributes have some intrinsic utility, and that when I offer you a good or bundle of goods, you have to aggregate the utility from the attributes, including substitution in some complicated way. That's a very engineering way to think about it. I understand it's not a Canara way to think about it. So we're thinking, oh, well, so where do these attribute value maps live? Are they like the visual cortex? Can we learn how they aggregate? Can we learn how substitution is physically implemented as engineers? And one of the predictions of this is that as I move around in the environment, if the same mechanisms operate in value cortex, and we have every reason to believe they do, then the attribute value maps anneal in the same way from experiences that you have as you move through the environment getting rewards. And so the temporal and spatial and, you can think about axes like saltiness and sugar or mating or things like that. And so we've often talked about, Michael Platt and I have often talked about taking his monkeys at Cayo Santiago and putting a little camera on their head and tracking them from birth and trying to get the complete CAP-Z choice set, like every choice the monkey ever makes for 2 years. And then do this analysis on it and ask what would be the maximum information representation of the choice set. And you could imagine doing experiments like these guys do, where you change the structure of the choice set, of the life choice set, and ask, does it change the representation and does it change the choice? So, I mean, I think the answer is yes. And Aldo knows we're working
Do we
on...
have an estimate how big the effect is in the visual system that is much easier to understand?
If we take out one eye,
No,
all of
I mean,
that goes away.
If you
What do you
raise
mean?
two frogs, otherwise identical, in two environments in which, I don't know, the frequency of
Yeah,
the
that's-
distribution of the luminosity is completely different.
Yeah, so
Do
that's
you
a...
think you will see it?
Oh, yeah, definitely. And now that experiment has not been well done. It's
Has
been
not
done,
been-
it's been done in the corner solution. The corner solutions are clear,
Well, the-
and they're well predicted by the biochemistry, and you can simulate that problem. So if you buy that we can, that our models for the corner solutions are perfect, or near perfect at the biophysical level, then the answer is yes. We once talked about doing an experiment where we embedded a contact lens in the eye of a baby tadpole, right? And the idea would be to create some other pattern, but it has to be embedded right at the lensing point. It can't be in front or it won't. It actually, if you think it through, it's actually optically technical to do this. And we kind of abandoned the project once we discovered how hard it would be to build these miniature contact lenses and implant them in the eyes of growing tadpoles. But that's the right experiment for that system. Right? Another example of that would be if I could put a noise screen in front of the... Here's a good way to do it. You take the little froggy, you put him in an environment where somehow or other he's in a video arena and you're allowed to control the statistics of his visual world perfectly. Now it's hard because he can't move, or I mean it's a technically hard problem, but you ought to be able to make any structure you want.
Okay, but then it's much easier to change the stochastic nature of the reward process.
Mm-hmm.
That experiment could be done tomorrow.
Right, but the problem is we don't know what the we don't have this neural representation story down yet. When you and I have...
No, but at the behavioral level, you take two tadpoles completely identical.
Okay, so that's great, but now we're getting into this domain that you know that this is what I work on every day, which is the precision of rungs, and how the precision of rungs is a lawful feature of the choice set. And more recently, we've been talking about how it's the history of the choice set. So I agree with all of that. I mean, I think, but now we're just talking economics, Aldo. This is not helpful.
I think your point about the environmental variation is a very important one. And I 100% agree with it. An old example, of course, in human genetics is phenylketonuria. Is phenylketonuria a disorder of diet or a disorder of genetics? Well, it's obviously both. But you would never have figured out phenylketonuria if you tried to study people's diets, because there wasn't meaningful variation.
Mm-hmm.
So you only figured it out because you studied their genetics. Then you understood the importance of diet. So they're both important. The question is, what strategies are the right ones to take? And do heritability statistics suggest anything about what the more powerful strategies might be?
I agree. I think that's the $64,000 question.
I have a very simple question. The frog eye, the third eye, how much do you have to put in there? Do you have to, how much of the retinal nerve do you have to put it? I mean, how deep do you have to get the retinal nerve in the brain for it to actually...
Do you want to
find
do
its
this
way?
experiment?
And how do you test, how do you know if a frog is binocular? What's the experiment to show
Okay,
that?
so that's great. I don't know why you're interested in this. So the answer is frogs are different than us. Amphibians, if you whack off their digit or a foot, they grow it back. That's a really good feature that we don't have. So what you actually do is you take, so they're monocular, their eyes look to this side, so this eye projects to this brain. So what you actually, the best way to do it is you cut this nerve, so now this is denervated because you don't want it crystallized, you want it fluid. And you take the second eye and you stick it on pointing in a slightly different direction, but when you harvest it, you harvest out the whole optic nerve. And you lay it right next to the other optic nerve. You lay the two of them on the anterior border of the tectum, which is the natural growing endpoint where all the gradients are. And nothing happens for a few days while the tectum sort of settles back to its I have to grow a new eye mode. And then when it gets into that mode, you see the neurons take off, they send growth cones in, as I showed you. And then they do this assorted thing. So you have to do it this way. If you let it fully crystallize, you don't get necessarily as good a result. It works a little. So this goes to a question, and the biochemistry was worked out basically, so you can decrystallize it biochemically now. So this gets to this interesting issue which you're poking around on, which is of crystallization. And the general story seems to be that you don't ever fully crystallize the nervous system, that you change the rate. You want to think of it as changing the time constant in this story over which you're computing the covariance structure. And so you want to think that when we're young and we have no priors, we're willing to time average over a very short time scale to derive a rough estimate of the covariance structure. That gives us a visual system fast. And then you want to kind of anneal that, right? Because now you want to basically be continuously reducing ambiguity. That's basically what you're doing. This is like Knightian ambiguity annealing to risk. And so you have this nice smooth function, and it seems to inflect in humans around, for most brain areas, around 4-ish, 5 years. But for some of the brain systems, frontal cortical brain systems, that may be doing averaging over a much longer time scale, that doesn't start to anneal till like 18, 19. So I think there's a lot of heterogeneity in brain systems on it.
I think we could talk about
Yeah,
this for 2
let's-
hours.
let's
It's
quit.
absolutely
I agree.
fascinating, but I'm very hardwired on schedule. So
I'm
I think we want to just
delighted.
thank you right now. Thank you.